Hello everyone, welcome to our channel. As we know, human beings are made up of billions of cells, and a proper coordination among all these cells is essential for our living and various specific function to be carried out inside our body. So, to understand all this coordination and intercellular communication, we are going to discuss about the cellular activation, in which we will discuss about the cell signaling, signal transduction pathways, and the transcription factor. So intercellular communication is essential for multicellular organism, as we have discussed. So extracellular signals determine whether a cell lives, die, or remain in a quiescent stage or perform a specific function. This all decided by an extracellular signal. Intercellular signaling is critical in developing embryo, tissue organization, and tissues to act in an appropriate concert. Loss of intercellular signaling seen in or result in unregulated growth, which is seen in cancers, or an extrinsic stress like condition, which is seen in shock. So first is cell signaling. Signals that most cells respond to can be classified into several groups. First is danger and pathogens. They may act as a signal, like damaged cells and microbes stimulate certain cells to respond accordingly and to eliminate these microbes and to deal with the danger or necrotic cell or their products. Second signal is cell-cell contact, which is seen in or carried out by adhesion molecule and gap junctions. Third type of signaling is cell extracellular matrix contact. This is carried out by integrins. And fourth type of signaling is secreted molecules. For example, growth factors, cytokines, and hormones play an important role in this kind of signaling. So signaling is there are four kinds of signaling: danger and pathogen mediated, cell-cell contact, cell extracellular matrix contact, and signaling by the secreted molecules. Signaling pathways can also be classified based on the spatial relationship between sending and receiving cell. So based on a spatial relationship between sending and receiving cell, signaling pathways can be a paracrine signaling, in which cells in the vicinity are affected, like this cell secrete the various material and they act on the cells on the vicinity. Second is autocrine signaling, in which the secreted material act on the same cell. This is seen during the development or positive negative feedback in the various hormones or other cellular mediators secretion. Third kind of signaling is synaptic signaling, which is seen at specific sites known as synapses and carried out by the neurotransmitters. Fourth signaling pathway is endocrine signaling, in which mediator or hormone acts on a distant site. Now we will briefly understand how this all signaling works. First, a signal, or also known as a ligand, which can be a growth factor, hormone, microbe, etc. They react with a signal receptor, which can be intracellular or present on the cell surface. After the interaction of this ligand and its receptor, there are activation of signal transduction pathways and result into the transcription. And this transcription affect the expression or repression of gene and result into a desired action. Signaling pathways bind their respective receptors and initiate a cascade of intracellular events and ultimately result into a desired cellular response. These ligands usually have high affinity for receptors and bind with exquisite specificity with these receptors. Receptors can be cell surface receptor or intracellular receptor. Intracellular receptors are activated by lipid soluble ligand. Lipid soluble ligand can cross the plasma membrane and directly reach or bind with the intracellular receptor. This is seen with the vitamin D or steroid hormones. Endothelial cells secrete nitric oxide, which diffuse to adjacent smooth muscle cell and activate the guanyl cyclase, which produce cyclic GMP and result into the smooth muscle relaxation. This is how endothelial cell control the vasomotor tone. Now in cell surface receptor, they are transmembrane receptor with an extracellular domain that bind with the ligand or signal. Depending on the receptor ligand binding, there is activation of various mechanisms. There can be opening of ion channels, or their receptor ligand binding can activate GTP binding protein. This can activate an enzyme like tyrosine kinase, or receptor ligand complex can trigger a proteolytic event or change protein binding or stability, and ultimately result into the activation of transcription factor. G protein coupled receptor and tyrosine kinase associated receptor, they usually involve in the cellular proliferation. Proteolytic or conformational changes in receptors, they usually involve in the normal development, which is seen with Notch, WNT and Hetke of pathways. After cell signaling, now we will discuss about the signal transduction pathways. So a ligand or signal react with a cell surface receptor and result into a receptor cross-linking or physical changes in the receptor structure. Conformational changes, which leads to the conformational changes in the cytosolic tail of receptor and activate the intracellular biochemical events. Cellular receptors are grouped into several types based on the signaling mechanism and intracellular biochemical pathways. In receptor signaling, formation or modification of biochemical intermediates and activation of enzymes, which result into the active transcription factor. Active transcription factor enter into the nucleus and alter the pattern of gene expression. Receptor, now we will discuss about the various receptors. Receptor associated with the tyrosine kinase activity Downstream phosphorylation is a common pathway of signal transduction in receptor associated with kinase activity. Changes in receptor geometry stimulate enzymatic activity or recruited intracellular kinases. They add the charged phosphate residue to target molecules. Like tyrosine kinase add charged phosphate to tyrosine. Serine threonine kinase add charged phosphates to serine threonine, and lipid kinases add phosphate residue to lipid substrates. There are phosphatases. They remove the phosphate residue and usually inhibit the signal transduction. In receptor associated with kinase activity, we have two types of receptor. First is receptor tyrosine kinases. They are an integral membrane protein. Ligand induced cross-linking activate tyrosine kinase domain, which is located on the cytosolic tail. They act as a receptor for insulin, epidermal growth factor, and platelet-derived growth factor. Non-receptor tyrosine kinases, they act as a receptor for immune mediators, cytokines, and integrins. After binding with the ligand, non-receptor tyrosine kinases phosphorylate specific motifs on the receptor or other proteins. SRC, a cellular homologue of the transforming protein of the Rous sarcoma virus. SRC family kinases, they are also non-receptor tyrosine kinases, contain unique functional region known as SRC homology or SH domain. SH2 domain bind to receptor and phosphorylated by other kinases and result into the aggregation of multiple enzymes. SH2 domain mediate protein-protein interaction, which is rich in proline-rich sequences. Non-receptor tyrosine kinase based receptor, they react with the ligand and with the help of non-receptor tyrosine kinases activate the various intracellular pathways. In receptor tyrosine kinase, ligand act with the receptor tyrosine kinase and result into the phosphorylation, downstream phosphorylation, and further activation of the signal transduction pathways. So this is an example of receptor tyrosine kinase pathway, a growth factor received by a growth factor receptor. After this interaction between the growth factor and growth factor receptor, there is a receptor dimerization and autophosphorylation of the tyrosine residue. This will result into the bridging protein couples the receptor to inactive GDP bound RAS. So this is a bridging protein, and after the phosphorylation of tyrosine, this bridging protein bind this inactive RAS with this tyrosine kinase receptor. Their binding will result into the activation of the RAS. Inactive RAS is GDP binding, and after phosphorylation, this will convert into an active RAS, which is GTP binding RAS. Active RAS stimulate RAF, and which further stimulate the MAP kinase and result into the activation of the transcription, and on the other hand RAS can also activate phosphoinositol kinase, and which also activate the transcription. Activation of transcription through the MYC protein will result into the cell cycle progression. So this is an example of how our receptor tyrosine kinase works after attaching with the appropriate ligand. Activated RAS eventually hydrolyzes GTP to GDP and converting RAS to its inactive form. Mutation of the RAS may result into delayed GTP hydrolysis and will result into an augmented proliferative signaling or increased cell proliferation. Now another type of receptors are G protein coupled receptors. They are 7 transmembrane or serpentine receptor because they cross the plasma membrane 7 times, that's why also known as 7 transmembrane or serpentine receptor. More than 1500 such receptors have been identified. After interaction with the ligand, G protein coupled receptor activate a G protein which is inactive with the GTP, and after phosphorylation converting into an active G protein which is GTP binding protein. G protein coupled receptor and ligand complex further activate the cyclic AMP and inositol triphosphate. Inositol triphosphate will release the calcium from endoplasmic reticulum and responsible for the various actions. Now another category of receptor is nuclear receptor. As we have discussed earlier, lipid soluble ligand directly cross the plasma cell membrane and bind their receptor. This ligand receptor complex directly bind with the nuclear DNA and activate or repress the gene transcription. Other classes of receptors are important in embryonic development and cell fate determination and functioning of mature cell within the immune system. Rather than enzymatic activity, they rely on protein-protein interaction to transduce the signal. Receptor proteins for Notch family and WNT family are come in this category. Notch ligand react with the Notch receptor, and via proteolytic cleavage of the Notch receptor there is a nuclear translocation of the cytoplasmic piece of Notch receptor, and after the nuclear translocation there is a formation of transcription complex. In WNT protein ligand react with the Frizzled family receptor, which is a type of G protein coupled receptor. This complex stabilizes beta catenin and translocate beta catenin to the nucleus and result into the formation of transcription complex. Normally beta catenin degraded by ubiquitin proteasomal degradation complex. WNT and Frizzled receptor complex activate the intracellular proteins which degrade this degradation targeting complex for the beta catenin and will result into the stabilization of beta catenin. Now we will discuss about the transcription factor. Signal transduction pathway modulate the gene transcription and ultimately result into the effect of the cellular function. Transcription factor that regulate cell division are product of several growth promoting genes such as MYC and JUN, and the cell cycle inhibiting genes such as P53. So this is all about the cellular interaction or cellular activation, how a signal react with the receptor and will result into the various activation of various signal transduction pathway. Ultimately they activate a transcription factor which either express or suppress a gene expression and result into a desired action. Thank you.